My name is Stefanie Weidtkamp-Peters and I'm heading the imaging facility at Düsseldorf University, what is called Center for Advanced Imaging. We are, as a core facility, we have many collaborations with different research groups. And what people are interested in to look for protein interaction and also for dynamic behavior of protein and other components within their different kind of samples. So we are looking at very different kind of samples. We have plant people doing research in every kind of plant tissue. We have people from the medical faculty and they study tissue and also living cells. And of course we have the normal cell biologists and they come with their cell culture samples to us. We have two core areas in our facility. We do a lot of spectroscopy, so we do film imaging. So we are looking for fat, that means we look for homo fat and hetero fat to tell you something about the interaction of proteins in great detail. And we also do super resolution. And there we want to go for studying super resolution in living cells and dynamic processes. Interested in doing spectroscopy in living cells. So what we want to do is working with weak expression levels of our fluorescent proteins, for example. So we have weak signals. On the other hand, to do proper spectroscopy, we need to collect a lot of photons. So what we need from a microscope system is best sensitivity we can get. So for us it's very, very important that all the optical parts in the system are almost perfect and that we can really squeeze out every photon that is available from our sample. That's most important for us. Two steps, let's say, because we need to bring in somehow the fluorescence in our samples, because we need the fluorescence as a reporter for us. And there are different ways, and as a facility, of course, I mentioned that we have many kinds of different samples, so plant and cell culture, tissue, whatever. So we are, as a facility, helping our customers, the other research groups, in preparing this kind of fluorescent samples. And once it is in, the fluorescence, and of course it's a critical step to move from where the sample is grown, where it's cultured, for example, to the microscope. This should be a very short distance, and if it's mounted on the microscope, we need an ambient environment for the cells, for example, to stay alive. But then there's a problem. You want to avoid any interference with your microscopy system. So you need good conditions for your cells, for example, but you also need good conditions for imaging. So that's challenging, but yeah, you have to work on it to get proper results in the end. It is very important to have proper conditions for your cells, to keep everything constant, to make your results comparable between different measurement dates, because you want to see changes in your cells happening not due to the environment, but due to the things happening in your cells, or if you look for a mutant or whatever. So for this reason, you have to rely on the environment, on everything keeping stable, and then you can trust your results in the end. We like the Olympus 60 times 1.2 water immersion lens a lot, because it's a very nice objective for spectroscopy. And of course detectors are very important. And on the other hand, as we sometimes want to modify our system to bring in our own detectors, we need flexibility. So we need access to incoupling of our lasers, of different lasers, for example. And also on the other side, the detection, we need flexibility to put in our own detectors. And there we just need also flexibility in the system to do it or to arrange it how we need it for our purpose.